there's bottom.
Yep. My altitude's
All
still
right, we've
40.
got
I'll
bottom
keep
in
coming
sight.
down.
All right, so we've got bottom in sight. On first glance, it looks like it's moderately sedimented. It looks more, yeah, it looks more like we're looking at sediment and maybe some
my sonar back to a normal
out of place blocks of rock.
configuration
It's quite possible it might be indurated. It might be fairly cemented. It's
looks
hard to
pretty
know.
cemented, I think.
Yeah, it looks like it's cemented. I would agree.
And there's not very much biology in sight.
Is there a sponge?
We can see, yeah, a couple sponges, but we're still quite a way off bottom, about 15 meters.
again?
Yeah,
so
we're just
maybe as we
eyeballing
get
from here.
closer,
Keep it on low or medium?
we'll see some more. But so far,
Thank you, Brian. That was
not very
awesome.
much.
Low's fine.
Okay.
What happened to my event log? There it is. Okay.
Oh,
But
put back the black as the background.
there, how's that? Better. Okay. Now, shore-based scientists, just because you can't see anything doesn't mean we're not going to make you participate today. So I hope you are ready to do some interpretive biology and geology from shore based entirely on description. How about it?
Let's keep coming down.
Silence.
It's a bamboo.
That's a darn good guess, Chris. 80% of the time.
So I'm seeing uphill more off to my port side, which agrees with HIPAC that we're just off to the side of the ridge.
Okay.
Looks like it, although the depths are still off.
Now they're making it really hard. Now, no, not even any audio descriptions.
So as we
Sorry,
go
I'm
along,
typing
I'll
in the
try to
event log.
figure exactly where we are on the high bed.
So what we are seeing, and this is based, again, we're not yet exactly at the
So do you
bottom.
want me
We're
to
just
turn
slowly
more
moving
towards
into
the
it. So we're
uphill
looking
first?
at this. It looks
Yeah,
like
that'd
there
be
are some
fine.
large
You want me to lateral
manganese-coated
over?
crusts,
Just kinda like figuring this
clasts, and it looks like it looks a little bit like a scree slope, but just based on observation alone, it does look like it's cemented in place. So we'll have to find out as we move towards things. And so we see at least one sponge, which I think Diva mentioned. But again, we're not there yet. We're not totally set up. So we're just sort of eyeballing around at the moment.
So our first sponge looks like a glass sponge, perhaps from the family Pheronematidae.
I'm going to go ahead and change our Baylite plant to northwest if y'all are still okay with that.
Maybe
Sounds
Sounds
good.
good.
a polyopogon.
Bridge nav.
Really?
Definitely take all these
I was
ideas
gonna
with
say,
a pinch of salt.
You're
We
going to
are
make me
on
write all
bottom.
these
We're gonna
down? I
update
don't
our
even know how to
Baylite
pronounce
plan
these.
to,
Look, his
I know. I'm looking over your shoulder.
to about 310
Possibly-
degrees.
Polyopogon.
Pogon.
Oh,
Good
Chris
stuff.
already
Good
has it in.
combo.
Thank you, Chris. That's probably going to be a lot easier than me attempting to type.
Yeah, maybe just a little.
Okay. I'll come
Before
Yeah.
this cruise, I would have told you my spelling was quite good. And now I would no longer believe that. All right.
Do you think this looks almost like carbonate?
I mean, it could be, like, in terms of the cementation, you think?
Yeah, like
Yeah, I would.
It's just
I could totally believe that. It's hard to know, of course, when
Yeah,
you're not.
yeah.
Unless you take a sample of something, especially when you're talking about rocks. Unless you really, really see that perfect lithology, you don't know what you're looking at.
And the deck department will be power washing and acid washing today on the bow as well as the boat deck. So please stay aware when you head outside. Thank you.
For those of you following along at home, today is power washing day. Great. Shore-based scientists, I should mention that we are going to continue to do the audio stream like we're actually transmitting offshore, because we don't know if the feeds might come back at any time, and we do want to be able to, if we can get a replay out later, we'd still like to have audio discussion. So we will probably still be asking you general questions, even if we can't ask you to describe what you're seeing at the moment. Is that okay?
Sounds like a good plan.
Good. And because I've just said that, Scott, you're on the line, right?
He's like, no.
Can you tell us something just general and interesting about the corals we might expect to see today?
Sure. So one of the things that we are interested in overall is the diversity and abundance of these corals. And most of what we see on these seamounts at these locations are octocorals, so more generally known as sea fans and soft corals. There are some stony corals, but we don't see them as much, or we haven't around here. We may see some cup corals and some very small colonies of the stony corals. But octocorals are usually quite abundant, as I say, sea fans. Why are we interested in the diversity and the abundance? So we can learn a few different things about the environment we're in. If we see lots and lots of corals, we may learn that... I'm sorry, it sounds like the video's trying to come through. I just had to turn that down.
Is the ship
I
underway?
apologize.
Okay.
Negative.
Okay.
So if we see, for example,
Just
high
not getting set
abundances
up.
of corals, that might tell us that there's quite a lot of food in this area, or that the currents are appropriate for
come wide.
sessile fauna, those animals
Come
that
wide,
live
Ron.
attached to the bottom for living there. So that's a very interesting place if we're thinking about conservation and places we might want to protect. In terms of the biodiversity, it's essentially how many
Roland,
different kinds
we weren't
of
able to
corals,
finish our white
or
balance
fish,
today
or snails, or
because of
crabs,
some
or shrimp,
hydraulic
any of these
issues.
animals. How many
Do you
different
wanna
species are there?
Use your
We've had some
try
good
to
looks in other places in the
manually
ocean. Last
do the
fall, we
best you
spent a
can.
lot of time around Hawaii in the
I was
northwest
starting to play
Hawaiian
with
Islands,
it, but
and we saw a range of species. And we now would
yeah,
like to ask,
I was
well,
just
do we
flipping
see
through
the
those.
same
There's
species
nothing
over
that
here, or do we see a whole
looked
new set of
obviously
species?
better, but
If we see a whole new set of
I'm going to come
species,
down a
this
bit
tells
and
us
push out further.
essentially
Copy.
that different regions of the ocean, even though they're connected by water, may have their own
wanna
regional fauna, their own
Try a
regional
little white
species.
balance on that.
And if
spine.
we are interested as a society to protect the particular species, then we might need to think about, well, maybe we need to protect this part of the ocean and that part of the ocean as separate entities instead of just considering the entire Pacific as one big ocean. So it doesn't matter if we do something in one place versus another, because those animals over there will eventually come back here. So the more we know about the specific diversity in an area, the better we can think about how we want to use the resources in an area, how we want to protect them, and so on.
Sure, you want me to
In the
sit
deep
down to get a
sea,
nice
the most common sorts of octocorals that we
We're gonna get our sample
encounter
pilot.
belong to a few families. So you've heard us talk a lot about the bamboo corals.
There's also a
family
white on the
Acididae.
upper right,
It's a very
maybe a bigger source.
diverse family, lots of different species and genera,
Copy
and
that.
many that we haven't yet described, but we are in the act of doing so. Very
Yeah, that
often,
one's a lot
as Deb and Diva will do, they'll ask us for our help in identifying them.
Okay,
And
Pilot,
we
I
do
have
our
you at
best,
40
but it's
degrees
really
in my
difficult to identify
Okay,
without
I'm
actually
just gonna
having a
move out
sample, especially when you're
Copy.
exploring a new area. One of the ways that these
But we
sets of
can move at
cruises
least
are extremely valuable
20
is
meters
that we
to
get
get
this
closer in.
stunning imagery,
Depending
maybe
on
not right this
maybe
second,
even
but
30.
we will have
we
this
wanna wait
stunning
there.
imagery coming off of the ship.
I think so.
And where we're fortunate
just
enough
go
to
ahead.
get a collection, then we can
Can we
compare
zoom on the sponge
what we
pilot,
have in
please?
our
Ah, yes.
hands,
We're
what
actually
we
gonna
see in the
white
video,
balance on the
so that the
sponge.
next time we see it on video,
we can get
then
a
we
25
can
meter
provide
move in.
a better,
Okay, yeah.
better
Figure
identification.
130.
So bamboo
That's
corals,
good
another
positioning.
group of
One,
sea fans, the octocorals that we commonly
What do you
see are the Primnoidae. They're very cool because they have these,
Yeah, if you're
small
on
skeletal
board with
structures
it.
that look like little suits of
Yeah,
armor.
that sounds good.
Also extremely
Uh,
diverse,
what
more
speed
diverse
should we do?
in
Should we
fact
keep it
than
kinda low,
bamboo coral.
0.2?
Then
0.2,
there's
yeah.
the Chrysogorgiid corals.
Also,
They have,
just so I
kind
have
of
this
iridescent
updated and
gold
can update
skeleton.
people as needed,
Again, some
are we going to be
beautiful
cutting
shapes
this
and
guy's show
sizes.
today?
Polyps,
to-
small
unknown.
those
Yeah,
the feeding
Jim,
structure of the
unknown.
colony
Okay.
look like little teardrops and so on.
Bridge,
Before I
Nav.
go on, I'm
Sorry
going
about
to
that,
take
Shoreside, we believe the feeds may be back. How do you see the streams back on shore?
Can we get a 25 meter move 130 degrees bearing, 0.2 knots.
Did anyone hear Roland? Do you guys
I did not
have
hear
video feeds?
If you do, you should be looking at a
Okay, copy.
polyopogon sponge.
Up and down.
Do not see a video feed.
Something happened. I just put it on the small monitor just to save the video wall with frozen video. Let me go turn it on again, but the scene on the small monitor is different. I think it's frozen now at a different place.
Hey, Chris, we're actually getting
that might
reports
too,
that the high speed might have just gone back up, so let us know if you get that video.
Yep. For all of you, please let us know if the video starts kicking in. Oh, cute! Look at that.
So this is likely a polyopogon sponge with a tiny ophiuroid commensal living on it.
Super tiny.
But the thing that's striking me most, apart from this beautiful sponge we're seeing, is that the geology here is unlike both of the seamounts that we visited before. You know, we thought today's dive, yesterday's dive, and the day before would be quite similar. And
Yes.
what we're finding is that none have been like each other.
But abundant. No, that was a joke. It's actually very true. This looks more like
Just going for
just
color.
poorly sorted, poorly sorted clasts in what looks like it's probably some kind of cemented, possibly carbonate, although I'm not willing to really say that, crust.
You can
It is
lose the,
not particularly beautiful
orange
geology at this time.
sponge.
But I mean, on 2 days ago, we had very thick
Manganese
manganese
crust,
crust.
yeah.
You know, almost
This-
looks a little rounded.
This still has manganese crust on it, but you're right, it doesn't have that botryoidal
Yeah.
texture. It doesn't have those round, pebbly shapes at the moment.
And
The
then
way
If we
it's
move up, we
sponge.
may see that.
And then yesterday were these very dramatic sort of cliff faces
With pillow basalt.
with pillow basalts, yeah,
my favorite.
which also had a manganese crust. But
Yes,
but
but-
hey, this is
So far, it's different. We'll have to see as we move up what we are, what we're taking a look
I
at.
believe that's exactly
Pilot, do you have a
what
moment?
you do from a sponge.
Okay.
Are
Going to turn the
Can you hear
video
me?
back over to Dan.
no.
Okay, watch leads.
Oh, there we go.
We're all set. What would you like to do?
Okay.
Okay, so the first thing we'd like to make sure of is that we're at the top of the ridge right now.
We came just slightly off to the side. We're facing east, and we think just one move will get us there.
Okay,
But we're in a
so
fairly flat. Waypoint 1 was in a fairly flat spot too, so it might not be very defined.
okay, that's good to know. So what we'd like to do is
I think I started creeping.
attain the top of the ridge and make sure we know where it is, and then we'd like to turn towards, I believe it's the south,
Yes.
and just start moving along.
2-3 minutes.
Okay. All right. Well, Sirius is on the way. We'll head east until we come to the local high, and then turn south.
Okay, great. Thank you so much. So for those of you who are watching at shore and for our shore-based scientists, we're actually going to move to the east a little bit. Our main track line for this particular geo was from north to south, so we're going to move east. We're going to try to find the top of the ridge feature that we'd like to move along. We want to make sure that we're up there at the top of the ridge because we expect that that's where the highest concentration of suspension-feeding
Do a 30
organisms is going to be.
25.
Then we will turn to the south, and as we move along the
Oh, you're
ridge,
talking to me.
we will try to follow
Nav's got you.
the highest density of organisms.
Forgot I changed.
What's that thing in the back?
So it looks like another sponge, quite a distance off in the background. But I don't think we've seen any corals so far.
Well, we haven't seen very much so far.
Yeah, but normally when we touch down, there's a bit more happening, and
Bridge,
it doesn't
Nav.
seem to be that way so far.
Yeah.
As Deb said, we haven't moved very much yet, but
We go ahead
our
and
pilots
extend
are
the
oriented
end
now,
of this
and
move by another 15
we're ready
meters.
to set off.
Yep.
Okay, a couple more sponges coming into view.
So we're
Yes.
at a depth of 1927, 1,927 meters.
I'll do my best to keep the foreground from getting really hot.
Is that from the upper lights?
Yeah.
And this is really interesting. I mean, this is very, very poorly sorted. we're looking at things that look like talus. There's a lot of angularity, but the size distribution of these rocks is really quite large.
Am I wrong in saying that they look a little metallic? Yes.
I don't really know what you mean by metallic.
Just the coloration.
Yes,
Okay.
I would. I mean,
Just figured I'd throw that out there.
are you saying? Do you feel like they look a little more orange than
yeah,
usual? Is
exactly.
that what you're trying to say?
Yeah.
So I'm not sure if that has to do with our imagery today or if that's real.
Yeah, watch leads. Our white balance mechanism is a little different today since
Okay.
our hydraulic systems aren't
Right.
functioning properly. We had to just white balance on that sponge we were just looking at.
Yeah. So does that mean
Can
that
we zoom
this
on this
is not
tiny
quite?
red animal pilot
yes.
directly in the center of the screen, please?
Diva never lets me finish.
Sorry. It's just I didn't want him to pass it.
Teacher, Diva never lets me finish.
Yeah. So I think this may be our first Anthomastus coral, or first coral at all.
But look
for
at all the
this,
sponges in the
for
background.
this dive. And there are a lot more sponges around. And there's also what looks like a chrysogorgid coming into view.
All right, so we're going to zoom on this coral. And which one was
Okay,
this,
video.
Diva?
Zooming in,
Can I have codes?
Anthomastus.
holding here.
Be a beautiful little Anthomastus right here.
We see co-pilot. There's
If you're looking
black
at an
balance.
Anthomastus and then you have a Chrysogorgia, this is a nice place to point out the variation in the way octocorals are constructed.
Flare
Anthomastus
and knee and gain.
essentially has small calcium carbonate
I'll check them.
skeletal elements that are all cemented in there together.
Do
It
you
looks
wanna
like a
get in
little
closer?
toadstool
Zooming
with
in.
polyps sticking out. Whereas
Yep.
the Chrysogorgia, it should have an
Holding
internal
here.
skeleton like a tree or a bush with the polyps on it.
Excellent. And so, so what family is this Anthomastus in? Or is that the wrong question? I'm trying to figure out what code to put in to the event log, Scott.
I would put in Coro, C-O-R-O, Anthomastus.
Thank you.
Is that
And you're
full
totally
video?
right. It definitely
little
does look
more.
like it's a little bit
Yeah,
like a
it's
toadstool. It has a
Actually,
bottom
it is
piece,
full.
and then
Yep.
it has
Sorry.
these nice coral
on the
sort of
edge.
branching out, polyps branching out from it.
With 4 tentacles per polyp.
No.
Octocoral. I mean 8, sorry.
I'm like, no,
8.
that's more than four.
Sorry.
I'm glad you made that correction. 'Cause I'm getting very excited about what
Back
I'm
to
going
the
to see.
count.
Scott was like,
Sorry, Scott. That was me
No
testing you,
species.
whether you were listening or not.
all right, pilot.
Very good.
Pilot, I think we're ready to move on
Zoom
to
out.
if there's a chrysogorgid in the area.
There was one just behind.
Full wide.
For if
Another
you
difference you'll see between
They
these
look like
two
little
is that the
bushes.
polyps and the Anthomastus can completely retract into the tissue.
Yeah, so you can see it just at the top of the screen.
They can never completely retract because the
a slightly white patch to the left of the center
polyps are always just hanging
on a rock,
Oh, yeah.
center of the
Yeah,
screen right
I
now.
see it now.
Yeah.
All right, so as we zoom in on this chrysogorgid, I have to read
Great
one
job.
of the comments that Bruce Mundy, who's one of our fisheries experts in Hawaii, said. He
Hey,
said,
are we on
we're sort of
location?
like the Bathel and
Thank you.
abyssal fishes, seeing light but no images. And so I think that's very funny.
I don't know, guy was a little bit off.
But we're working on it, Bruce.
I thought this was just a white patch on a rock. Okay, video.
Zooming in.
So we're
Whoops,
zooming
I
in
just
on this
stepped out for a
chrysogorgid.
moment and just came back to hear my
Holding.
name.
Oh, we were just reading your comment and enjoying it.
Yep, so this is definitely a chrysogorgid. I don't see any commensal squat lobsters as I usually have.
Coming
I don't
up.
see any commensals, as a matter of fact.
Well, you shouldn't be moving anymore, are you?
And is this bushy all the way to the rock, or is
Zoom
this
in a little bit more
planar, or does this look like a dandelion?
Full zoom.
It's
It's pretty bushy.
Bushy.
So it's probably in the genus Chrysogorgia.
Yes, I would agree with that.
It's very pretty and it's moving.
Some really informative comments
I know,
coming
that
your
was
way.
so
I'm sure.
helpful. This is what happens when you ask the geology. Describe it. Actually, I'm going to go back to my question for the pilot. So, pilot, may I ask you a question?
Yes.
So we never seem to get it. I'm confused about the answer about is the imagery a little orangey today, or is that what it really looks like?
Oh, maybe you didn't hear video before. Yeah, The color balance that we usually do at about 1,000 meters depth is on the hydraulic manipulator. So we couldn't do that on the way down.
Right.
So it could be a little orangey, yes.
Okay, perfect. Thank you.
Scott, just flipping through the ID guide, and it's definitely a Chrysogorgia.
Okay. Zooming out.
All right. Is that correct video?
Yep. That was correct.
No, yeah.
All right, so as we move along, we're seeing a few different types of a few different sponges. I don't know if they're the same type or not.
No, they do
Diva?
all look quite... Oh, no, well, so there are two different ones in view right now.
Two different sponges?
Oh, no, three different ones in view right now.
Where? I see one in the center. I've got that one. Thank you.
And then there's one to the left and then one in the background.
Those are all sponges?
Yes.
Okay.
So the one in the front looks like a polyopogon, or some type of ferronematidae. It's very sort of circular, disc-like, quite large. And you can see a lot of spicule detail.
You want to shoot the lasers at it to see how big it is?
Sure.
Pilot, can we get the lasers on?
Yes.
Now the one to the left looks like a Tridopleura, sort of leaf-shaped. I'm pretty sure it is a Tridopleura.
There's lasers on this guy.
Perfect.
Do you want to do a quick zoom on there?
Yeah, a quick zoom would be great.
Of the other two, is there one you'd like to zoom on?
Thank you so much. Which of the others would you like to zoom on, Diva? Perfect. That's awesome.
Yeah,
Thank
that's
you.
beautiful. Thank you, pilots.
Yeah, I'm probably settled out now, and it looks like
Whenever you're clear video.
it's harder for me to tell where the local high is here.
Can we do the one in the background? Would that be okay?
Yeah, copy. It's not.
And there's
matching
some
up
very
exactly.
large out-of-place
Of
rocks
course.
here.
So it's hard for me to
Can,
tell you
and
either.
before we zoom on the one in the background, can we zoom on this animal on the rock, just in the
Yeah,
foreground?
the spot lobster.
I think it might be a squat lobster, maybe.
I do love a squat
you are
lobster.
where I
Yeah,
think
we
you are,
all
though,
do.
You're right in the middle of two 10-meter contours, so it's like in every direction you shouldn't see anything.
Well, we can keep coming this way. Maybe another 30,
Okay.
20.
Yeah. 25?
Yeah, that's fine. Yeah.
Okay. Bridge nav. Can we get a ship move of 25 meters bearing 130?
So we're
0.02
currently
notches.
approaching what looks like a farreid sponge, but it's slightly off color, so I think it may be dead or dying. But in the foreground, we've got a beautiful squat lobster, which is actually upside down. Yeah.
It is upside down. And then what is it on?
And it's living on a quite large bunch of hydroid corals. And then,
Tilting up.
pilots, can we get a zoom on the little blobs on the rock just below?
Sure. Video whenever you're clear of this shot. I'll
They
tilt
might
down.
be sponges.
It's a hydroid coral.
Okay.
Thank you.
Yeah, so we're also seeing some very small white blobs on the rock face, and I can't quite. Can we pan right, please?
Yeah, there's more over there. I should probably get moving soon, right, for the pilot?
I think you're okay. We just started that ship move.
There we go.
They look like young barnacles, not the stalked ones. Oh yeah, they definitely are young barnacles. One of them has their feeding cirri extended, and they're likely from the family Balanoidea. Balanoidea. My apologies, Balanoidea.
Looking
Okay,
at
that's great,
70.
pilots. Thank you. If we could get a zoom on the sponge behind the farreid sponge, that would be great. Thank you.
Hop over there.
I think you're fine.
So we are continuing to move mainly to the east.
Copy.
We are at a depth of 1922 right now.
Understood.
Yeah, that doesn't look like a healthy sponge.
No, it definitely looks a little off-colored, but it's been colonized by what looks like quite a large comatulid crinoid.
Is it okay to say video whenever you're clear then? Because that's what I usually say.
Yeah, that's what I try to say.
Not to blame you, but
Yeah.
So this farreid looks a little bit like Aspidoscapulia.
we can definitely hold things longer. Yeah.
Okay, there's
Set up for him earlier.
lasers on it. Co-pilot want to get lasers off.
Sure. Lasers off.
Okay, video, you're free to zoom.
Okay, hold here for a second. And zooming in. And I will hold here.
And it's got a commensal shrimp living on it, perhaps from the family, from the genus Labeus.
And you're okay, pilot. I'm looking down 80.
Okay, it looks like you're on pretty flat terrain.
Right, I looked out ahead.
Sorry,
It
you
seems
said
like we're
shrimp?
okay.
Shrimp, yeah. Labeus, Lebbeus. And there was also a comatulid crinoid. I didn't get to see how many
that's
arms
great.
it had yet.
We'll zoom out.
Let's go down to
I
the
will
crinoid.
hold partial.
Okay, I'll go down here. Okay, you can go in on the crinoid.
Zooming in.
Don't forget
Holding.
video. I have my lower two that I can throw on. If you don't think the
Are these just the uppers?
Yeah.
I think it's got 9 arms.
we can always try the lowers.
This
Why don't we,
commensal
yeah, put 'em
comatulid
on just to see
crinoid.
what it looks like.
Okay.
And it's also yellow brown,
Brown.
I guess.
Didn't really do much, huh?
Didn't do much, but we'll keep them on.
Okay, great. Thank you,
Okay.
pilots.
All right. Anything else you want, video?
I think that'll do. Zooming out.
Come out. You can push ahead a little.
All right.
Pilot. And make
Full
me more
wide.
comfortable.
Thank you. Yeah, those lowers don't seem to cause a problem.
Copy.
Okay, we're more than happy to set off in the direction we're supposed to be heading.
We're actually already moving that way.
Oh, great. On top of things as usual.
So, I guess I'll head uphill this way until there's no more uphill, which should be soon, and then probably turn starboard.
Yeah, turn southwest. I imagine it'll be up. I think I can kind of see the top here.
So as we're flying on, we're beginning to get some slightly larger
Maybe not.
rock fragments,
This
no,
is the
no.
top.
some of which are
Same
quite
here.
angular, some of which are quite rounded,
Yeah, maybe.
all covered in what looks like a manganese
So
crust.
I just
It's gonna
And
be a
in the
little flat right
distance,
here.
The sponge is up
Interestingly, no corals, not one coral,
Our 10
Which is
meters
strange.
takes place
but
over
quite a
120
couple
meters on this one.
sponges, a lot of the large polyopagons it looks like.
say I'm
Saw
on the
Crescent
top.
Gorge,
Oh,
right?
can we zoom in on this,
It's coral.
a sea star on top of that sponge, just about to go
Yes.
off screen?
Of course.
What do you think, Nav? That's probably the top, huh?
I think so.
Okay.
Like I was saying, it's a really long distance for the contour change at this beginning point.
Yeah. Flat. It's pretty flat down here. So
Yeah,
where did that?
I would think we'd want to turn a little further south if that, but this high pack could easily be off.
How far are we into that ship move? We're done.
We're done. We're done with that. We stopped a couple minutes ago. You should actually be moving right now.
Yeah. I
Okay,
think we
video?
should, next move. Let's try to
Ready?
move south. I think.
Zooming in.
And
Okay,
so
how
we've
about
got
just
another
straight up
dead
south at
sponge
180?
in front of us that looks like one of these
Yeah,
holding here.
Triopleuras,
I think
but it
that'd be
also
okay.
has again a commensal
Another
shrimp
25er?
living on it, and then there's also a
Yeah,
brisingid
let's do
on the
that.
top of it.
Bridge now.
Zooming in.
Let's go ahead and put in a 25 meter move, bearing 180, 0.2 knots.
Could you put
Okay,
in
copy.
shrimp and brisingid for me, please?
Okay, video is
I put
clear.
ASR for asteroid,
I
but
am
I don't know if
aligned
there's
on
anything
heading.
more. Just
Pretty
check
good.
the.
Zooming out.
We've got a photo bomber up to the left.
Full wide.
You come up.
Thank you. Watch the question for you.
Go
We
ahead.
see a lot of these kind of large brown sponges that look dead.
Uh-huh.
But I can't say I've seen many live ones that are that large and type. Am I just forgetting them, or?
Sorry, wait, so repeat that question?
I
Sorry.
can't say I've seen many of that type of sponge totally alive and white and healthy looking.
On this dive or in general?
Just in general.
And that's an interesting point.
We see a lot of, or I feel like we see a lot of them that aren't alive.
So of that, what, Faraday or something like that, or Faraday?
No, that one's treated pleura, I think.
Oh, okay.
So here's so
There are many more
another one of
sponges in
the
this picture.
Faradays below there that also doesn't look very healthy. Oh, there's a synaphobranchid swimming down there. Can we get a quick zoom on that, please?
Yes. Is the ship still going?
We're going 180 and I'm aligned.
Siri, you're going to have to
You're
spell
good.
synaphobranchid.
Coming down to it.
So, shore-based scientists, there's a reason that I don't generally type in words like synaphobranchid, because I can't spell them. But luckily, Diva's helping. So all of a sudden, we're in these really big
video.
class, big rocks, completely out of place. It looked like these actually look like they have a lot less manganese sediment on them, manganese crust.
Okay, so the synaphobranchid looks like it's got its lateral line of dots going down its body.
It doesn't look very chewed up the way that some of the other ones we've seen have been.
Doesn't.
You know what I mean? Is that true or not?
Yeah, definitely. It's definitely synaphobranchidae, but I'm not sure whether it's a synaphobranchus or an Iliophis. What are the main distinguishing features, Bruce?
Okay. First, if the lateral line pores that you're seeing, the dots along the side of the body, if they're pale or almost whitish, that would be Iliophis.
No, these ones don't look whitish. These
I
ones,
see black. Is that
yeah,
what you're
no,
looking
these
at?
ones don't look. They definitely look black or grey.
You want lasers? Briefly?
Not yet.
Okay. The Iliophis tend to have a more slender body, slender head, and a kind of a rounded snout. The Synaphobranchus are a little deeper bodied, often brown or charcoal gray, and the snout's a little more pointed. Also, the Synaphobranchus have a peculiar way of swimming, so that the back part of the anal fin along the lower part of the body has a, sort of an indentation toward the tail, and then the tail slightly curved up that makes it look like they've got a rounded tail fin, a rounded caudal fin, instead of the typical eel body that goes back straight.
Okay, that's helpful. I'm going to say this is Synaphobranchus brevidorsalis, but I mean, you can review the video later and tell me how well I did on my fish IDs.
Okay, that sounds good. And the dorsal fin position is another one. Synaphobranchus brevidorsalis has a dorsal fin that begins. It starts about halfway back on the body, or maybe a third of the way back on the body. And the Iliophis, the dorsal fin goes up almost all the way to the head.
No, so it was definitely a Synaphobranchus then.
Cool. All right, so I believe we're still moving along. Go ahead, Bruce. Sorry.
Let's see what's out there.
Well, I was going to say other Synaphobranchus
right now,
have the dorsal fin that's a little further forward than the brevidorsalis, but not all the way up to the head.
Okay, so we've moved away
And
from the
it's still
fish
on
now,
mode, just so you
and we're continuing on.
Field of
And
sponges.
in our field of view, we can see about 10 sponges, some of which are quite large, but again, no corals.
Not yet. But we've seen a few, just
I mean,
far in
like
between.
2 or 3, but
Right.
nothing like where we've been on these geodes before and we see fields of coral.
But it was very interesting. Remember, Scott was telling us yesterday that what they've seen in some places is sort of a zonation, where we see sponges further down and then corals as we move higher up the ridge. What's that thing to the back right? Is that also a sponge?
Yep, everything we're seeing right now is sponges.
Any different types?
Yes, so there's what looks like 3 Pheronematidae sponges. And then in the back, there's another one of the large.
turn
Okay.
10 degrees, keep you in view.
That sounds good. I think I'm
Wow,
mostly
that's a
uphill,
beautiful
so-
sponge.
Can we have the lasers on just quickly so we can gauge how big it is, please?
I'm holding.
Sponge people, this is a really big looking sponge and very attractive, which I know helps a lot. Are those spicules at the bottom, Diva?
That's nice. All right,
Yes.
you can throw 'em
So that's one of the, I'm going to guess,
Okay, so
identifying
it's-
features of these
1,
polyophagons.
2, 3,
You want 'em up?
maybe 40 centimeters across.
And then about
And then
70
1, 2, yeah, around
high.
70 meters high. Oh, 70 centimeters. Centimeters, not meters.
on. Yeah, you can turn 'em off.
Centimeters.
Thank you.
Right.
And zooming in.
So not big as far as, you know, what we've seen on
hold
past
here.
expeditions, but big
Tilt up
big
a
for
little.
this expedition, definitely.
That's good. Thank you.
Am I leading the direction of motion?
And this one looks really healthy. You can see this attachment spicules at
Maybe
the
a
bottom.
centimeter
Yeah.
clear.
We can
All right.
kill the lowers if they're
Zoom in.
reflecting, too.
I think it's okay for now. It's full zoom.
What a beautiful
Beautiful.
pattern. So we zoomed in on it, and you really can see that mesh-like pattern very well.
Pilot, can you come down a couple meters?
Affirmative. Starting those
Awesome.
heaves again.
Navigator, this is watch lead.
Zooming out. Hold it here.
Go ahead, watch lead.
All right. Yeah,
So
nice.
am I correct in assuming that we've sort of gotten to what we think is the top of the ridge and we're going to start moving mostly south?
Yes.
Okay, perfect. Thank you.
And if that changes, I'll let you know.
Okay, zooming out.
Excellent. Thanks a lot. Have we already put in a ship move for it?
Yes, we've been moving. In fact, we're moving
Full
right
wide.
now.
Excellent. Great.
Okay, watch lead on the other sponges.
There was a
Nearby.
tall branching one off to the right, please.
Pan to
Yes.
the right.
There we go. That one
Oh, is
in
there
the
a dead
in the
sponge in front of it?
No, yeah, so not the
in the
dead
back.
one,
Right.
the one in the back.
No, I was just mentioning there was a dead sponge in front of it.
Yeah, so 222 seems to be pretty good.
So for those of you who are following along, I really hope that Scott is not correct that the Telecon is also struggling a little bit, because that's going to make us really sad here on the ship. We are very much enjoying hearing what you guys have to say about the general interests and the characteristics of what we're looking at. What's the oop?
Yeah, okay, I think we're turning around. Never mind.
Yeah, I just wanted to get
Yeah.
the good side.
Great, thank you.
At least what I hope is the good side.
So I turn to follow you, and I'm going to translate a little, but
Okay.
I think I'm going to hold this heading for the next move anyway.
All right.
You looking uphill there, co-pilot?
More or less, yeah.
So I'm unsure about the
so,
ID
yeah.
of this one. It's either Faraday, Aspidoscapulia,
The depths
or
are off. I don't. Like 20 meters.
Euretidae, Cornelas matinee. It's just
Yeah.
got that tall branching morphology. We're lower on the stalk. There are some small sort of extensions coming off of it, and those get bigger as you move up the sponge.
Okay, video.
Zooming in.
Nav, maybe every half hour, 20 minutes or so. I guess we're only on for a little bit more, but just drop a target with like a reference
For
depth.
yeah, okay.
How does that sound?
Can you put in the log that it's either Faraday,
I did put
this,
in Faraday.
Like in
or,
parentheses?
but I'm not sure
Like it with
about
a
my
dimension
ID,
or something?
Oh, okay.
or this.
Full
How's that?
zoom.
Yeah, that's fine.
Or Eurydice?
Yep.
How does the next word go?
Cornelas matinee.
Look at you. Did you take Latin as a child, Diva?
No.
Wow.
I took English and bad English.
Okay, zooming out. Get a full shot.
Thank you, pilot.
And tilt down a bit.
Are we ready to move on?
Yes, we are. Thank you.
All right,
Yeah,
we are
once
ready
that
to move.
180 move is done, if it isn't already.
It
we-
should be.
so we
Zoom
can
out
get
a bit
a
more. Hold there.
220 in.
That's great.
220. Okay.
Yeah.
25 again.
There's a little shrimp or something heading into
I
it.
think we can do 30, yeah.
Ooh, okay.
Where's a shrimp?
Good job.
There's
Some
something
little
floating, but I don't know what
creature.
it is.
Okay.
Looks
can
like
zoom on the
Bridge
creature.
nav.
It's tiny.
Flock.
Yeah, go for it. I think it's actually going past.
Oh, there.
Hey,
yeah,
let's
I
do
see
that
what you
30
mean.
meter ship move, bearing 220,
You all
0.2 knots,
clear in
please.
this, video?
What's that?
You all clear?
Yeah. I'll come full wide. Thank you.
Good copy.
Okay.
So we just got a ship move in for you.
All right. What's the bearing? 220.
Okay, so I think we've seen all of these sponges already.
Yep.
So if we could,
That's where I am. I'm just bouncing around.
Want to start moving? Yep. All right, so pilot, as it works for you, we'd like to kind of try to keep moving a little bit
We are on our way.
until we tell you to zoom on something else.
Southwest. Southwest.
We should watch these at watch these at this speed. We still have time to zoom on things.
Perfect, thank you. I was just making a joke.
Oh. But we're not going to stop the ship, I don't think,
Okay,
unless something crazy happens.
different sponge, same sponge. We're seeing many sponges. There's got to be 10 in the frame right now,
or more.
or more.
And they're all this sort of polyopogon shape.
Even the one in the front.
Getting out there a little
Yeah.
Yeah, I stopped here. I'll turn back across.
bit. I can come down a little bit. I don't want to.
Chris is going to
Right into
review
the
this
tether there.
imagery after and be like, what was Diva talking about?
No, I agree. These are all sponges.
Thanks, Violet, for that vote of confidence.
Even I thought they were sponges, so really, it's the next step of what is this sponge that we're trying to get to. So, which actually, knowing our ROV team, they probably also know that. So I think what we're doing is, as we're moving south, we're sort of panning to the left and the right, trying to see what's on this ridge so that we can figure out, once we start seeing that higher density of organisms, what we can follow.
Which is interesting because there's very little else, it appears, other than sponges here. And the sponges aren't that frequent.
And they're pretty frequent.
I mean, it's not like...
They're not super
It's
high
not
density.
a huge abundance, exactly. And there's not very high diversity either, it looks like. Can we ever zoom on that little vase-like one just about to dip off screen? Is that
Yeah.
possible?
Yep, it's a little far
Please?
up there, but I'll...
Sorry.
Yeah,
can do.
you gotta
And
get
it's
a tug,
interesting
I think.
because we had a lot of conversations
Well,
about how
turn.
if you have
I don't
hard
wanna- I'm not
substrate, you're
coming
more
down
likely to
anymore.
have corals and sponges, right?
That's okay. I'll get
And
it
we
from
are
there.
now seeing a lot more hard substrate.
video,
We're
let's
seeing a
zoom on
lot
this
more class.
base, kind
We're
of
seeing a
far
lot
away.
more rocks. It
zooming in.
definitely looks manganese coated.
Hold
Oh,
in
we're
here.
starting to see that manganese texture.
So we're definitely looking at a Euplectellidae sponge right now. An absolutely beautiful specimen. As well as, what are all these thin white stalks around it?
That's a good question. That is a gorgeous sponge. These
It
are
is,
beautiful
isn't it?
today.
Yeah.
I mean, not that the sponges aren't always beautiful, Chris and Shirley. They're always quite attractive.
So it's definitely Euplectellidae, perhaps Corbitella,
So what do you think those little thin
or
streamy things are?
Dictylus.
What are those? The little thin things.
We're going to need to get a bit of a better
Gotta
zoom.
turn to keep you in view.
Okay, I'll
10
just get
degrees.
this and I'll shoot back.
Okay.
Try to hold it.
For those of you who, shoreside scientists who are reading the event log right now, I'm just going to apologize in advance because I'm logging while Diva's talking, and as you can imagine,
Okay, video.
I'm trying my best to
Zoom?
keep up,
Zooming in.
but it's not working that well. So
Gorgeous
please
sponge.
bear
with
me.
me.
Gorgeous
Full zoom.
sponge.
Okay. Try to hold the top.
Absolutely beautiful.
now might be a good time to just ask: Are people able to hear the telecon?
I didn't quite catch what you asked there.
Can you hear me?
Was that meant? Were you being
Usually,
facetious?
except when you ask that question.
Okay, as long as we're
Pilots,
having
can
some.
we have a zoom on the white stalky things, please?
Zooming out.
Just to the
Holding
so
here
just
partial.
to the around
Yeah.
there, exactly.
Okay, video, you want to go on the base? There's two
Can you
white
help me ID
stalks.
this?
Zooming in.
Hard to ID.
Holding. Full zoom.
What is
Sponge.
that?
So I think they're cladorizers.
The little tiny skinny things?
Yeah. So
Yeah,
also
so
what we've
shot
started to
is
notice
different.
that there are some just single stalked sponges, very thin, and they look quite a lot like
Happy with that video?
a Bestipluma.
Zoom out.
So just a single stalk with single, just sponge.
Oh,
yeah.
okay.
Full wide.
So just a single stalk with sort of radiations coming off of them,
Hello, pilot.
like yeah, I don't know how to describe it.
I will take your word for that because I have to tell you, it looked
Off a
like
little
just a
and
tiny
back.
little
They
stringy
definitely
thing.
look like
Yeah, I'll come back to
cladorizers.
TTs.
Okay.
Can you put cladorizers
Yes,
in?
I can
please?
try. Clad.
Here.
Oh, perfect. Clado.
So again, I had hopes that they would be corals, but no. So far, all we're seeing are sponges of various descriptions, glass and otherwise.
And so I believe we're still moving to the south.
Probably near the
We're
end of
still
this
kind
move.
of panning back and
With the
forth,
ship anyway.
taking a look.
Probably.
I have to tell you, things are very exciting in the control room today.
Yeah,
We've
there's
got
a lot going on.
A lot going on, even though, I realize that for you guys there's not a lot going on, but we're hoping to fix that. But there is definitely a lot of activity here trying to make sure that we can get these feeds to shore and that you can continue to hear us on the telecon. So rest assured, we're trying to get it done.
So, Copilot and Nav, just to pass it on, the comp's at 23%, and it's been holding for, I think, about an hour. Nav's been making notes.
Thank you, Bridge. That's the end of the move.
Great. So if it definitely keep a close
And for
eye
those
on
of
it,
you who
yeah,
are following
plug
along with us
falls
today,
again.
we're having some technical difficulties
Think about
here,
what to do.
but we are still having a
Yep.
really interesting dive so far on Delcano Seamount, which is a
Geo.
I was about to say it's a Cretaceous age geo. Thanks for jumping in there. And of course, Diva, would you like to tell us what a
no,
geo is?
I'm really sorry. So it's a flat-topped seamount.
Therefore, Delcano Seamount is technically correct.
Definitely.
But yes, it is a geo, and because it's so old, Cretaceous age, between 65 million and 145 million years old, we expect to see a fairly heavy manganese crust on these rocks. Interestingly enough, so far we haven't seen too much of that very characteristic manganese, texture, that botryoidal texture we were expecting. But I'm pretty sure there's a pretty heavy crust here. What we've also been seeing as we've been moving up and then along the ridge are lots and lots of sponges. Well, I think there's lots and lots of sponges. Diva doesn't think they're quite as abundant as I do. And she's a biologist, so she's probably right. But we haven't seen very many corals yet. So we are going to continue on as the ship moves. We will follow along with that, and we will see what we can see. And then we will do a terrible job of describing it. And hopefully you will correct us. And right now we're actually just waiting here for a moment while there is an ROV team shift. And we are at a depth of 1,917 meters. For the shore-based scientists, I am trying to type that in every so often because, as you've probably noticed, we also don't have a nav string coming into the event log. And that is because that's another shoreside service. So we're not getting that right now. At least that's my understanding. Perhaps I should go ask.
All right, watch lead. This is Josh on pilot. And,
Awesome.
Ready to go. whatever you are.
Josh, we've been waiting for you all day.
I know. Hey, Josh.
So, what we'd like to do, Josh, is just continue to move in the southward direction. We want to follow along this ridge. And what we'd like to do is sort of pan left and right, looking to see where the greatest density of organisms is, and then follow it.
Roger that.
Thank you. And we have already seen a fair number of sponges that we've zoomed in on, so we may ask to try to zoom on some things that aren't sponges if we find them.
Yeah, copy that.
Great.
Good morning, Josh.
Good morning. How's it going, video?
It's going all right.
So we're just starting to move along
Notice
again.
that the colors aren't going to be quite.
We are looking at manganese encrusted,
today.
somewhat angular looking
able to
rocks,
finish
actually.
it.
These are a
Yeah,
little
quite.
more angular than they were before.
Terrible.
Something to keep
Kind of
in mind.
craggy.
Yeah.
Do you notice?
We did the
What's
best
that
we
down
could
in
on
the
a white
bottom?
sponge.
Well, we're just about to go off screen. Was that a crinoid?
I think it was a dead sponge with a crinoid
Does it
on it.
look like you?
Okay, thank you.
Well, I mean,
It's a little red. And
it's a good
orange,
opportunity to just like go for it, just adjust on the fly.
yeah, which
And
we have been doing.
for those of you
Yeah.
who are either shore-based and
Turn the
didn't hear the
saturation way
beginning
up.
of the dive or are
full
following
art.
along right now, the ROV had
it
lost
art-
its
is it
hydraulics
capital R?
about halfway down
Capital A
today,
art?
so we do not have any sampling capabilities today.
Just off to my starboard.
Let's zoom in here.
And it also means it affects the lighting just a little bit because
here.
there are many lights that are on swing arms on this ROV, and the swing arms don't work when the hydraulics don't work. So today we won't be sampling, but we will still be collecting great imagery.
So we're currently looking at another one of
So we're
the
currently
farreid
not moving.
sponges that, again, does not
the
look
ship
very
anyway.
healthy.
Or probably-
It's got
probably to
a
stop with the
probably labea
series as
shrimp
well.
in it, as well as a comatulid crinoid,
All right,
perhaps from the family Antidonidae.
let's come in tight real quick.
Okay, zooming in
Something red on there. Oh,
and
it's a
holding
little
here.
So I heard a crinoid. What else?
A shrimp, likely from the family genus Labeus. Sorry.
Labeus. And Patty Fryer, one of our geologists on shore, who will actually be the geology
Okay,
science
when you're
lead
happy.
on leg 3
going
of
just
this expedition,
seconds longer.
is very
Nice
happy to
job.
hear that there are rocks. Patty, there are rocks.
You guys ready for a ship move? Or?
Yes, there are
Let's
rocks.
look in the sonar.
Okay, that's great.
And another
Zoom
one of
out.
those
Yeah,
cladorized sponges
go
off to
30
the
meter
left.
hop.
I think those cladorizeds are actually quite
wide.
abundant, but
What
because
direction?
they're a bit smaller,
was too
we
You don't
haven't
really
been
see them as
noticing
much?
them. Yeah.
It's like
You
it's
mean on
more
this
to
particular
starboard.
dive, not in
Yeah,
general, right?
Yeah.
Yeah, earlier they were going more like 180.
So we're just going up this ridge. to try to
It's
find them.
abundant,
Yeah.
but not very diverse.
So I have to explain this joke because I think this is really funny.
Pilots,
So
just wanna let
we
you know
read
I've
an
had a
article
request from the editors
yesterday that will
we
not
try
be
to
named.
hold our
And,
nice shots a little longer.
One of the lines in it said that here at the seafloor for this
cutting those
Marianas
short,
expedition,
and it's been harder
there
to
was
edit
abundant
around, so
geology, although
be
it wasn't
conscious
very
effort of
diverse. And
holding those
we've been
nicer
making
ones
jokes
a
about
few
that
seconds
for
more.
at least the last
Yeah,
12
copy.
hours. Because, as all of you know, there is
What does it look
geology on the
like?
seafloor. It's what makes up the
thinking
seafloor.
Globally, we're going to need to go south, right?
Yeah.
So therefore, it's very
maybe
abundant.
Like 240.
And it
Always.
actually has been diverse.
On D2.
Perhaps that person just
Yeah.
watched too many of our pillow lava dives. Abundant geology.
Even if we just moved to where D2 is now, it would be a better direction, right?
Yeah, I mean, looking at the sonar, the strongest return. I mean, that might be just what you're closest to, but the strongest return is you're heading right about now.
It's chilly in here today.
So like 240.
Okay, well that's more left from where we were. So we'll go 30 meters, 240.
Sure.
Yeah.
Oh, I just realized I can't see the high pack with imagery. Okay, thank you. Yeah, I don't want to...
Bridge nav.
Okay.
Like a move, please. 30 meters,
Fine.
bearing 240.2 knots. Thank you.
Let's come in partial video.
Zoom in partial. Holding here.
Just staining.
So these are pretty angular clasts. They are coated in manganese crust, and they actually have a lot of what we've seen. We've seen this on previous dives. It's just sort of a flocculent or stuff.
Zooming in.
That's the best word I can come up with.
On the rocks right now?
On the rocks. It's that sort of, when you pick them up and they're sort of a little bit hairy, but it's probably not really.
So sometimes that's actually hydroids growing on them.
Yeah, but
as
these
well,
don't look
but
like they're
little-
like
no.
super hydroidy.
But that sponge, not very holding in place very well.
There you go. So you think
I
that's
can't-
a cladoresid
yes,
sponge?
So 100% cladoresid
Oh, now I
sponge.
see its little
yeah,
tiny
you can see its
spikes coming out of the sides.
extensions coming off of it.
Yeah, I'm trying to give you some here.
That's right. That's better.
So you can type in that genus, perhaps.
You can come in any tighter.
Asbestos.
That's full.
Asbestopluma.
It's a small guy.
Put a question mark.
Okay. Diva and I are sharing a keyboard today. So I'm the
Deb is the scribe.
designated typist.
Scribe.
Scribe sounds better.
Yeah,
Thank
zooming
you.
out. Oop. Full wide.
Thank you. A lot of bouncing today, huh?
Yeah.
Watch lead nav.
You go ahead.
Let's come in on this guy.
I'm not hearing you, I don't think.
Can you hear us now?
Yeah.
Okay.
I see waypoint
Holding
2. Is that our destination for the
Yeah,
day?
you can come in tighter.
Zooming in.
I don't think so, but if you
Holding
zoom
here.
out, do we see anything else?
No, I tried that and didn't.
Oh. Then perhaps it is. How far away is it?
250 meters?
No, it's definitely not our
Yeah,
waypoint
then
for
that
the
can't
day.
be right.
Okay.
It's supposed
What
to be
depth were we supposed
past.
to be at?
We're supposed to go over the high and then dip down a little bit
Okay.
further south.
Yeah, but I think, let me,
Oh,
let me see if I can find the
maybe I
data.
didn't look.
Yeah,
It's possible I didn't look quite far enough because I just went
Yeah,
to the
I
high
think
point.
we didn't, because Andy
There
and
it
I,
is. No,
popped it in
no,
last
no.
night.
So yeah, there's no waypoint set here for
Huh.
for a destination past the high point.
Yeah, I think something is wrong. So what we're trying to do is achieve the highest point of this first summit. So I
Yeah.
think the one that you're zooming in on at the bottom right now, the 1840.
Right.
I think that's what we'd like to try to achieve today if we can.
Yeah. Okay.
So the goal will be to just
Which
keep
is
moving.
750 meters from there. Yeah, we need to we
so-
need to boogie.
Yeah, so we're probably not going to get there, but even if we can start getting up towards the 1860
Okay.
contour, that would be great.
Okay. Well, I'll just try to keep us moving.
Great, thank you.
Yeah, we can just keep continuous, like,
Yeah. Yeah.
I think that's
40
exactly.
meter moves.
Yeah, I think that's exactly what we'd like to do because especially because we won't be stopping to sample. So if you can just keep us moving, and then at the time when you have to tell us that you need to keep moving and we have to stop zooming, that's totally cool.
Let's come in
Yeah.
partial.
Oh, internet's coming back, yeah.
Zooming in
Yay!
partial.
Wow, you're way
holding
too
here.
excited about
So
that.
my guess is we want to go more
How can
like
I check
180
Facebook
the
right
next,
now?
The next move, but
Says she who's looking at
Be
Twitter
helpful to-
even as even as the internet comes back.
Yeah, bathymetry makes it look like we should be heading straight south.
right.
Okay, come on out.
Zooming out.
Yeah, the ship is just about to the
Full wide.
end of this move, so I'd like to keep it moving.
Yeah, go ahead.
180. Does that sound?
It's going to take us sort of.
So supposedly we were
parallel to
on
the
both
wall.
the ITV and BBC News
Slope,
tonight.
But that's fine.
Awesome.
So
Parallel? You mean up the slope or
no, it's going to take
We
us
were
parallel.
or
so
are?
if you look
Oh, we
at
don't want to do that.
Where
it's
it
like
happened?
180.
Yeah.
We want to go up slope. Okay.
And
Okay.
upslope is where Levi is facing.
230.
230. Okay. We will go 230 then.
Well done, Okeanos.
For what?
Being on the news in England.
So who is John Copley?
He was my old PhD supervisor. He's not
Oh.
old, but
I
he's
understand.
excellent at science communication.
Bridge nav.
That's great. So if you can
Looks
hear
like we're
us,
just finishing up.
feel free to
So
go
you can come down.
read the
Levi,
article
a
on
little
Like,
BBC
bit.
give you another
News.
move,
Oh,
please.
it was
If you
on
look at your
the
altitude,
actual
30
news on the
meters.
TV.
30 meters.
I'm sorry, so go watch the
230
article on BBC
decimal
News.
2 knots.
Yeah, you gotta keep
Boy,
an eye on
I
the
feel
television.
like
I
You
mean,
can't watch an article.
You can watch a segment.
Good copy. Thank you.
I feel like Diva is kind of being mean to me today.
No, I'm not.